Today we are going to see the fourth lesson of Unit 3, which is protein synthesis. But before we have directly go to today's lesson, let's recap what we have seen in the previous lesson. In the previous lesson, we have seen molecular genetics, which is very important in genetics, which is one aspect of the science genetics, which is about gene and genetic engineering and the importance of genetic engineering, and we have seen importance of this genetic engineering. Today we are going to see protein, how protein is synthesized, protein synthesis. Now it's your turn to define and write down protein synthesis. What's protein synthesis? What do you understand by the term protein synthesis? Well done. Protein synthesis is a biological process by which cells generate new proteins, essential for structure, function, and regulation within an organism. You know, proteins are very important in structural as well as for functioning of our cell and regulation within an organism. This process involves two key stages, transcription and translation. So the two stages of protein synthesis are transcription and translation. Let's see one by one. Now let's consider events of protein synthesis. One of the events in the protein synthesis is transcription. The DNA code of a protein is rewritten in a molecule of messenger RNA. So DNA code is rewritten in a molecule of messenger RNA. This rewriting of a code is known as transcription. Rewriting DNA code to messenger RNA. This is known as transcribing the code in the DNA into messenger RNA, transcription. So the messenger RNA travels from the nucleus through pores in the nuclear envelope to ribosome. So transcription occurs in the nucleus, and now messenger RNA have a code that's copied from DNA, written from DNA, and messenger RNA travels from the nucleus to that of ribosome, which is outside of the nucleus, which is one of the cytoplasmic organelle, ribosome, very important in protein synthesis. Free amino acids are carried from the cytoplasm to that of ribosomes. Free amino acids are carried from cytoplasm by a molecule known as transfer RNA. Now, the work of transfer RNA is here to transfer amino acid and carry amino acid from the cytoplasm, freely found on the cytoplasm, to that of the ribosome, which is one organelle in the cytoplasm itself. So transfer RNA transfers and carries amino acid, free amino acid, from the cytoplasm to that of ribosome. Now we have seen two main functions, three main events. One is transcription. DNA is rewritten, messenger RNA, that's known as transcription. And we have seen that messenger RNA moves from the nucleus, outside from the nucleus, to that of ribosome. The third event is free amino acids are carried by transfer RNA from the cytoplasm to that of ribosome. The other event is ribosome. What's the function of ribosome then? Ribosomes reads the messenger RNA code that's coded during transcription and assembles the amino acid. Amino acid, also free amino acid, have entered to that of ribosome. We do have free amino acid in the ribosome. We do have messenger RNA now. So the messenger RNA code is assembled by the amino acid carried by transfer RNA into a protein. So this process is known as translation. So generally speaking, there are two stages of protein synthesis. One is transcription and the other is translation. During transcription, DNA code is transcribed into messenger RNA. The code is rewritten to messenger RNA, and this process of transcription occurs where? It occurs in the nucleus. And the messenger RNA go to the outside nucleus and goes to ribosome. So the actual process of protein synthesis occurs in the ribosome, which is one cytoplasmic organelle. And the process by which messenger RNA and the code in the messenger RNA and the free amino acids that are transcribed, that are transferred by transfer RNA, are assembled and carried into actual protein is known as translation. So translation occurs in ribosome. Now let's proceed and see genetic code. What's genetic code? Genetic code is a set of rules by which the sequence of bases in DNA or RNA is translated into a sequence of amino acid in protein. This code is universal system used by almost all organisms, reflecting the shared evolutionary heritage of life. This code is triplets, universal triplet code. So the code contains three nitrogenous base, three nitrogenous base. There are two codons which are very important and you have to know. You need to know them. One is starter codon. Adenine, uracil, guanine codes for methionine. This is signal for the start of translation. During translation, this is the starter codon. Adenine, uracil, guanine, which is found in the messenger RNA, is a starter codon. Stop codon. There are three stop codons. Uracil, adenine, uracil, adenine, guanine. Uracil, guanine, adenine are the end of translation. They are known as stop codons. This genetic code is held in the DNA molecule. It's a sequence of bases in the nucleotide of DNA that make up a gene that codes for protein, and each amino acid in the protein is coded by triplets, a sequence of 3 bases. From this, a gene is a sequence of base triplet in a DNA molecule that carries the code for protein. So in a gene, we do have triplet code. As we have seen, it's universal code. The triplet thiamine, adenine, thiamine is the DNA for amino acid tyrosine in humans, a giant redwood tree, a bacterium, or in any other living organism. So this codon is in a triplet, thiamine, adenine, thiamine, 3 nitrogenous bases. Now let's proceed and see in detail the 2 stages of that we have seen, transcription and translation in detail. We have said that in protein synthesis we do have 2 stages. One stage is transcription and the second stage is translation. Let's first see transcription. Transcription is the first stage of protein synthesis, where the DNA code is read to produce messenger RNA. So the DNA code is rewritten to produce a messenger RNA molecule. The code is transferred and produce a messenger RNA. This process is take place in the nucleus. This process, the first stage of protein synthesis, occurs in the nucleus. So if you are asked, the first stage of protein synthesis occurs where it occurs, then the answer is in the nucleus. The second stage is translation. Translation is where messenger RNA is used to build a chain of amino acid that forms a protein. This is the actual process of protein synthesis, the actual process of protein. Messenger RNA has a code which is copied from during transcription from DNA, and this code is used to build a chain of amino acid. And you know that proteins are made up of amino acids, and these amino acids give us proteins. This process takes place in the ribosome. And ribosomal RNA and transfer RNA are part of protein synthesis. So the two stages, transcription and translation, occurs in two different sites in the cell. You know, our cell has a nucleus and cytoplasmic organelles like mitochondria, lysosome, ribosome, and other organelles. So transcription, which is the first stage of protein synthesis, occurs in the nucleus. Whereas translation, which is the second stage of protein synthesis, which is the actual protein synthesis, occurs in the ribosome. Generally speaking, if you are asked where protein synthesis occurs, the answer will be ribosome, because the actual process occurs in the ribosome. But if you are asked in a stage-wise, the first stage, which is translation, occurs in the nucleus, whereby DNA code is read by and rewritten by messenger RNA. Messenger RNA moves from the nucleus to that of ribosome, and also free amino acids in the nucleus will go by the help of transfer RNA and enter to the ribosome. So in the ribosome, we do have free amino acid, we do have also messenger RNA. On messenger RNA carries code. So messenger RNA code is used to build a chain of amino acid that forms a protein. So the second stage was the last stage of protein synthesis, occurs in the ribosome. Now ribosomes are made up of ribosomal RNA and the protein organized into a large and small subunits. Ribosomal RNA organizes into a large and small subunits. Within the ribosome, there are three sites that can be occupied by transfer RNA molecule called APE sites. In the ribosome, these events occur. The first two codons of messenger RNA enter to the ribosome, one event. Transfer RNA molecule with amino acid attached transfers the complementary anticode to the first two codons of the messenger RNA, bind to this codon. So the code that we have on messenger RNA, their anticodon will develop into here and bind bind in the second stage. This is transfer RNA that transfers the free amino acid to that of ribosome. A peptide bond forms between the amino acid carried by these two transfer RNA molecule, and the dipeptide is transferred to the transfer RNA in the A site. The ribosome moves along the messenger RNA by one codon, bringing the third codon into the ribosome. At the same time, the free transfer exits ribosome and transfer RNA with the dipeptide moves into the P site, second site, and transfer RNA complementary anticodon binds with the third codon, bringing its amino acid into position, and next to the second amino acid is produced. A peptide bond forms between the second and third amino acid. A ribosome moves along the messenger RNA by one codon and bringing the fourth messenger RNA codon into the ribosome, and the whole process is repeated until a stop codon is in a position. So a stop codon stops the process, and translation ceases or stops when stop codon comes into existence. How protein synthesis is different in prokaryotic cells. We have two types of cells, eukaryotic cell and prokaryotic cell. In the case of eukaryotic cell, we have true nucleus and membrane-bound organelles, whereas in prokaryotic cell, we don't have true nucleus and we don't have membrane-bound organelles. So prokaryotic cell do not have a nucleus. We have seen the first stage of the protein synthesis, which is transcription, occurs in the nucleus. So in prokaryotic cells, we don't have nucleus. So prokaryotic messenger doesn't need post-transcriptional process. So in prokaryotic organisms or cells that are prokaryotic cell, their protein synthesis doesn't require transcriptional process because we don't have nucleus. And transcription, translation are coupled. Messenger RNA can be translated by ribosomes at one end of its nucleus molecule while it is still being transcribed from DNA at the other end. So end-to-end process will occur without nucleus. In the case of eukaryotic cell, obviously transcription and translation are separate because we do have true nucleus in eukaryotic cell. Transcription occurs in the nucleus, translation occurs in the cytoplasm and in the ribosome. Eukaryotic messenger RNA are modified before leaving to the nucleus. These are examples of proteins our body synthesizes: structural protein, enzyme protein, peptide hormone, antigen, antibody, their example and their function. Structural, for example, collagen is one example. It is very important in building fibers of cartilage, which is very important. And keratin is used to build nails and feathers. Enzyme, ATP synthase, DNA helicase, which is very important in DNA replication, and ATP synthesis in ATP, synthesizing ATP, which is one very important molecule. The function of ATP synthase is producing ATP from ADP and from phosphate, whereas DNA helicase is used unwinding the double helix of DNA. You know DNA is double helix, and in the process of replication, there is unwinding process. This unwinding process is done by the help of this enzyme. So enzymes are proteins. Hormones like insulin, Adrenaline are very important. Insulin is used to control plasma glucose concentration, which is one very important hormone. Adrenaline is used to fight response during fighting. This adrenaline is released, this hormone is released. Antigen A on red cell determine blood group. Antigen CD4 allows binding of HIV to T lymphocytes, which is very important. Antibody A causes clothing of red cells with antigen A. HIV antibodies destroys some HIV antigens. This will bring us to the end of today's lesson. Goodbye until we meet in the next lesson.